Okay, good. All right, so it's 11 o'clock. I'm going to start like I did last time, reviewing what we went over last time so that, you can kind of know what's most important here. Although I've already quizzed you on some of this. So anyway, we started the last lecture talking about active transport. So two different types of active transport. One is direct active or primary active transport, and the other is indirect or secondary active transport. So this is your active transport, which are pumps. So they're called direct active because they're going to directly bind ATP and use that to move ions or molecules or whatever. We've got three different kinds of pumps. You've got your P-type, which is kind of the main type in mammalian cells. When we're ever talking about sodium potassium ATPase, a calcium pump, those are the main ones, I guess. They're P-type pumps. And then you've got two different kinds of pumps that are structurally very similar. So they put them together here, the F-type and the V-type. V-type, again, are mammalian. You're going to find them on, or I should say eukaryotic. You're going to find them on lysosomes. So they're going to acidify the inside of the lysosome to activate them. The F-type then, the F-type is more prokaryotic. So you see them in microbes, but also in your mitochondria and probably chloroplasts, I'm assuming, although I know not a lot about plants, frankly. It's never been my interest, but plants are cool. They're just not my interest. Okay, anyway, so those are two different types. And then the third type that's pretty important medically is your ABC type pump. And the reason it's important is because of the MDR. And you can look at that animation later if you want to. But the MDR is the multi-drug resistance protein, which in the danger medically is if it shows up in a cancer cell, the cancer cell pumps the chemotherapy out of the cell and makes it ineffective. So that's kind of an important pump for that reason. Also, here's kind of a weird thing. I think I said this last time. Cystic fibrosis transmembrane regulator is a chloride ion channel, but it's structurally related to the ATP pump. So it's probably descended evolutionarily from one of these type pumps, but it turned into a channel, which, how, sequences change over time, and that's probably where it came from. Okay, so here's our sodium potassium ATPase mechanism. You should know this. Basically, it's got the E1 and the E2. In the E1 conformation, it binds sodium on the inside and ATP. Then it phosphorylates itself, and it kind of absorbs the energy of that third phosphate and changes conformation. Then the sodium goes out, potassium comes in, it dephosphorylates itself, and it goes back to the E1 conformation. And you can't see this, you can't see me, but I'm like waving my hands as if you can. So I feel like a bit of a weirdo here. Not your problem. Okay. Indirect active now has to do with, again, I'm not crazy about your the pictures in your book, but basically you've got one active transport, which is producing a proton gradient here in this example. And then here's your inactive transport, or your secondary active transport over here, which is using that concentration gradient, which, remember, is stored energy, to move something else against its concentration gradient. Now, this can be a little bit confusing because, some co-transporters are active and some are passive. I think the only way to really get it is to kind of memorize the ones that are active. So if it's sodium glucose, sodium amino acid, sodium calcium exchanger, they're all active. The ones that are passive are sodium hydrogen. For some reason, the sodium hydrogen exchanger is not active, and neither is the chloride bicarbonate antiporter. Those are both just facilitated diffusion. There's no energy involved in those. All right, so that's your indirect active. And then this is an example of why you would want an indirect active instead of just a regular transporter. So if the concentration gradient is in the direction that the cell is going to be useful for the cell, you just need a transporter. So this is your GLUT, which is your glucose transporter, and glucose is high inside the cell, so it's just going out passively down its concentration gradient. On the other hand, on the apical side of this cell, this is a small intestinal cell with microvilli. At the lumen side of the intestine, the body really wants to soak up all the glucose it can. It doesn't want to lose energy through defecation, right? It wants to pull all that glucose in. So you've got an active transport over here to make sure you get all that in against its concentration gradient. Okay, glucose is going to be higher inside the cell. And that's being driven by your sodium potassium ATPase again. So again, the whole point is when the cell needs to get it against its gradient, it's going to use this. When it's going downhill, it'll just use the regular transporter. And then what is this? Oh, your sodium calcium exchanger is another important one. Especially important cardiac. We'll see this in cardiac when we get there. Depending on how much cardiac we do, I'm not sure. Anyway, calcium metabolism, extremely important. This is in pretty much every cell. What you need to know, what you need to know here is to decrease calcium in the cell, this should be micromolar, not millimolar. To decrease calcium in the cell, you've got 3 ways to do that: 2 pumps and your sodium calcium exchanger. And then once you've got those huge concentration gradients, it's easy for it to go back downhill. So we've got just channels to get it back into the cytosol here and here. And you've got 2 different calcium release channels on the ER. And plasma membrane channels, there are a lot of different ones. Okay, so now we're getting close to what we're going to be talking about today. So I'm going to slow down a little bit. Channels. So when you've got a concentration gradient, the quickest way to get something in is a channel, because it's basically just a regulated pore in the membrane, and when it opens, the ions come flying through. And this is typically for signal transduction reasons. So here, this is kind of like one subunit of a potassium channel, or one domain of a Volch-dependent calcium or sodium channel. And it's got 6 transmembrane regions. One of them is positively charged, and that's going to allow that channel to sense voltage and open when the membrane potential changes. They also have this P loop that goes between the S5 and S6, and that's going to be part of the pore. So that's the selectivity filter of the channel. It will determine what ion is allowed to go through.
Dr. Slish?
Yes.
I think for all I can see is still slide 27 on membrane transport, and I think you're talking about the calcium transport slide, so I don't know if it's...
Yeah, I'm on slide 30. I'm on slide 34 already. Let me...
Okay, I see it now. It was just stuck on the first slide that we were talking about.
Okay, let me ask you a question. I'm like displaying this on mine. Tell me, does it change or no? Is it changing?
I only see you right now.
You're seeing me. Okay. Well, I'm a good-looking fella, but that's not what we want to go with right now. Let's go to. Oh, there I am. All right, let's change my share. Oh, wait, no. Cancel that. Still kind of getting used to this Microsoft Teams. Okay. Copy. Place. Okay. So I'm not going to review what we already reviewed. Unfortunately, you didn't get to see the figures while I was reviewing it, but let's go to the new stuff so we don't fall behind. Okay, I'm doing channels. Thanks for telling me, by the way. Please, I really need feedback, because like I say, I can't see what's going on. All I can see is what I'm doing here.
Yeah, this time it looks different because it looked like you were on a presenter or not on presenter view last time. We were just seeing kind of like that I don't know
that
what to
one.
call it, but now yeah, the editing page, but now it looks like presenter view. So hopefully the slides will
Okay.
advance.
Okay, fingers crossed here. What is this? All right. Let's go to hide presenter view. Okay, so here's our channel. And you've got these, this is like one subunit of the voltage and potassium channel or one domain of the calcium and sodium channels. So here's that domain here. And in the sodium and calcium channels, they're all in one big protein. They're all lined up here. What's important here is your S4, which is the voltage sensor. It's got a positive charge every third amino acid. And this P loop here, which is going to form a channel in the center. So you can see the P loop of each of the different subunits or domains coming together. And there's the channel in the center of the pore in the center of the channel. And these the amino acids here in this P loop are going to be looking at the ions coming through, and they're only going to let the right ion through. So Boltzmann sodium channel will only let sodium through. Potassium, only potassium, same for calcium. Okay, so that's for our channel structure. Channel function now, let me see if I can change my share. I've got an animation here of the different types of channels. Okay, so let's, okay, can you see my channels? Here's your voltage spin.
Yes.
Here's your voltage spin channel. When you can see the voltage on the membrane is normally negative inside, you depolarize, the S4 segment pulls back, gate opens, and then sodium goes through. Voltage spin channel. And then you've got ligand gated channels. Here, this could be acetylcholine comes in, flips it, and then once the sodium starts coming through, that changes the membrane potential. So we'll see that today in our synapses. And then we've got metabotropic channels, which we'll see a lot in axon bulbs, actually, controlling how much neurotransmitter gets released during an action potential. So those are our 4 different types of channels. Stop sharing. Let's get back to, why are you doing this to me? Mm-hmm. All right, yeah, this isn't going to work. All right, I don't know. Now I know exactly what doesn't work here for me. How did I get here? Oh. All right, I'm not going to do that much anymore because that just gets me lost. Come on, man. This is going to be chapter 3, which I don't want. Yeah, I don't want chapter 3. Sorry, guys. It's just this is not working right. Okay, so you can probably see slide 34, right? Yes?
Yep.
Yes.
Do you see slide 35 now?
Yes.
Yep.
Okay. All right, let's stick with this then. All right, so here's our channels and how they work. All right. And you've got stretch gated channels and gap junction channels. All right, so let's get to the new stuff. All right, so membrane potentials. So where does our membrane potential come from? This comes from two things really: asymmetric distribution of the ions and selective permeability. So asymmetric distribution of ions, that comes back to our sodium potassium ATPase. You've got that. So we're talking here about excitable cells, mostly neurons, also skeletal muscle, cardiac. But you've got sodium potassium ATPase working in the membrane, always pumping sodium out and potassium in. So what happens then, you've got low internal sodium and high internal potassium. So what does that mean? That means that for each of these ions, there's going to be a driving force for it to go across the membrane. So you've got a chemical driving force for sodium to come in and a chemical driving force for potassium to go out. The chemical driving force is basically just because you've got a higher concentration of it on the outside, it's got a driving force to come in. Now, the thing that complicates that is it's a charged particle, which means that its movement is going to be controlled by that concentration gradient, but also by its charge. Because if it's positive on the inside of the cell, a positive ion is going to have a hard time getting in, right? Because it has to go against that charge gradient. Even if its concentration gradient is really strong, it can't go through because it's positive. So for each of these ions, there's going to be a point where it will go through the membrane and a point where it's going to stop based on its electronic gradient. So that's why we call this electrochemical gradient, because its movement is controlled both by its concentration and by the charge on the membrane. So how do we calculate this? Now, don't ever worry about memorizing an equation. I'm not going to ask you to do math in this class, but you want to be able to look at this and know what it's saying. Basically, what it's saying is for potassium over here. Can you see my cursor? Anybody?
Yes.
Okay, so for potassium, you've got 4 millimole of potassium on the outside, 126 on the inside. So that times, that's going to give you a negative number because it's a fraction here, times 61 gives you 92 millivolts. Now, what does that mean? That means that because of the concentration of potassium, it will keep going out of the cell until that cell's membrane potential gets to minus 92. Okay, if you can go more negative than that, it'll come into the cell against its concentration gradient. So that's its electrochemical equilibrium potential. At minus 2 millivolts, if you open all the potassium channels, it will be in equilibrium, okay, in and out. Sodium is the opposite, right? High outside, low inside. So its equilibrium potential is going to be the other side, plus 60 millivolts. Okay, so that means that at a negative membrane potential, it's going to have a driving force to come in. Once you go more positive to 60, it's got a driving force to go out. That's called the reversal potential. So this Nernst potential is also called the reversal potential because that's where it's at equilibrium. And what does that mean? That means if you have only sodium channels in a cell and you open them all, the potential on the membrane will be plus 60. If you have only potassium channels and you open them all, the membrane potential will be minus 92. That's what these things are telling you. That's where they're going to be in equilibrium. That's where they're more or less driving the membrane potential to. So, but as you know, in cells, we don't have just one ion. We've got multiple ions going at the same time. And that's where we get our Goldman equation. So if you look at this, let me do something real quick. If you look at this Goldman equation and you black out that much of it, it's basically the Nernst equation, right? It's the Nernst equation for potassium. Although we've added in another factor here, P, which is the permeability, which means how capable is the potassium to go through the membrane. So what our Goldman potential is, what our Goldman equation is doing here is adding up all the Nernst potentials, but relative to their permeability. So let's say we're in a cell where all the sodium channels are closed and all the potassium channels are open. In that case, we could say that, I'm just writing this now so we can look at it. If that was true, then PK would be much greater than PNA, right? So if you had all your potassium channels open and none of your sodium channels, PK would be much greater than PNA. So then PNA would be close to 0 compared to this, which would be close to 1. Their permeabilities, if you put PK over PNA, this would be close to 1 and this would be close to 0, which would mean that this wouldn't be contributing to the membrane potential anymore. The membrane potential would be controlled by this. And that's more or less what we're going to see here in our action potential. When the sodium channels are closed, the potassium is going to control the membrane potential. When the sodium channels open, it will control the membrane potential. So the membrane will swing towards it. So we'll see that again in a minute. But basically, this Goldman equation, what I want you to know about the Goldman equation, it's basically adding up all the Nernst potentials relative to the permeability of each ion. Okay. So we're looking at what the membrane potential will be based on which ions are able to go towards their equilibrium. Okay, so at rest, when the cells are at rest, the main channels that are open are potassium leak channels. So here's our cell at rest. You guys see this resting membrane potential minus 70?
Yes.
Okay, good. So in this, say we're looking at the membrane of an axon of a neuron. You've got voltage-dependent sodium channels, they're closed. Voltage-dependent potassium channels, they're closed. But you've got this potassium leak. So potassium's leaking out of the cell. And so the membrane potential is going to be closest to its Nernst potential. And remember, Nernst potential is minus 94 for potassium. And so our membrane potential ends up about minus 70. Because in addition to this leak potassium, you got a very, very small leak sodium current. So that's bringing you away from that equilibrium potential of potassium, a little bit more positive. They're balancing out at about minus 70. Okay, so that's your resting potential. Leak potassium, potassium coming out, and that's holding your membrane potential at minus 70. Does that make sense? Am I going too fast?
No, you're good.
Is that good? Okay. Everybody hanging on?
Yep.
Okay, I'll assume yes. All right, so let's talk about the action potential now. Once you start talking about the action potential, you're talking about your sodium channel kinetics, because the sodium channel is going to be controlling the action potential, basically. So your sodium channel can be in 3 different states. At rest, your sodium channel is going to be in this closed state, where the voltage gate is closed, but then we've got this other gate, an inactivation gate, which is open. And so these we call available, because these are capable of opening at this point. So in this state then, the closed state, this is where you're at during at rest of the neuron. Now, when the voltage changes, when you depolarize the cell, that's going to open this voltage gate. And then the sodium channel goes from closed to open. Once it's in this open channel, it's got a certain time factor before it's going to go to inactivated. So this is time. So you've depolarized the cell, and over a period of about, I'd say 1 to 2 milliseconds, the sodium channel is going to like flicker open and closed a few times, and then it's going to go into this inactive state. Once it's here, it's not going to open again, okay, because it's not controlled by the voltage gate, it's controlled by this inactivation particle, and it's going to stay in this state until you repolarize. So we've changed the voltage backwards. So now once the cell repolarizes, it's going to take a certain amount of time for that channel to go for that inactivation particle to come back out, and then the cell becomes available again. And that period of time, I don't talk about it in this class, but keep in mind when you do talk about neurons in other classes, that time period is your relative refractory period. Okay, you've got absolute refractory period and relative refractory period. In the absolute refractory period, at the peak of the action potential, all of your sodium channels are inactivated, so you can't get another action potential. Once the cell repolarizes, it takes a certain amount of time before those channels are available again. That's your relative refractory period. All right, not important for this class, but I'm just throwing that out there. All right, so let's get back to our action potential. So what's going to happen in our action potential is a stimulus is going to come and stimulate these sodium channels to open. So here at our rest state, we're at about -70, and then these get stimulated open, and they're going to go from closed to open. Once they do, sodium rushes in. Now at this point, it's the most permeable ion. Okay, all you have is your leak potassium, and that's not going to cut it. So your sodium is the most permeable ion. It comes flooding in, and the membrane potential goes up towards that reversal potential of sodium, towards the Nernst potential of sodium. Now, slightly before you reach that peak, though, right about here, your voltage-dependent potassium channels are going to sense that major depolarization, and they're going to open. And then, just at the peak, your voltage-dependent sodium channels are going to go from open to inactivated. So in this next phase of the action potential, then, these guys are inactivated, and these guys are open. So now we're going to rush towards the Nernst potential of potassium, which is like -90. So we rush down, and you can see we're down below where we were. This is the overshoot. So this is down below resting. And why is that? Remember, our resting potential is being controlled by the leak potassium, which is a really small current. Here we've got this huge potassium current, so we go even closer to the reversal potential of potassium, the Nernst potential of potassium. That's our overshoot. And then the next thing that's going to happen is once we've repolarized, these guys are going to close, and these got the voltage-dependent channels close again, and we go back to the situation where our leak potassium is holding our membrane potential here. So let me go over that again. I do have an animation, but I hate the thought of changing my share again, because every time I do that, I get messed up. But there are, if you pull up this this PowerPoint for yourself, you can see I've got an animation for this. I just don't feel like screwing around and getting lost again. Anyway, so at rest, leak potassium channels holding the membrane potential. At the peak of the action potential, you've got sodium rushing through, but these guys are inactivating, and these guys are about to open. In the repolarization and overshoot phase, this large potassium current is going out, and that's bringing you back towards the Nernst potential of potassium. And then those channels close, and you go back to rest. Okay, so that's your action potential. That's happening in the axon of neurons. Also in the plasma membrane of skeletal muscles, different for cardiac. We may or may not get to that. We'll see. Okay, any question about the action potential? Hello? Did I put you all to sleep with that? No questions. Okay. Okay, so that's one type of potential, which is your action potential. It's once it starts, it propagates all the way down the axon. We'll talk about that in a minute. The other type of potential is the graded potential. So the graded potential is what happens at synapses. So we're talking about axodendritic synapses, axosomatic. We're also talking about the neuromuscular junction, which came up last week. Anytime you've got a neuron interacting with an excitable cell, you've got yourself a synapse there, and we're going to see graded potentials there. And a graded potential is called graded because it's not all or nothing like an action potential. The more neurotransmitter that gets released, the stronger that potential is going to be. And typically the way this works is this presynaptic axon here will get a train of action potentials coming down, and it will release a series of quanta of neurotransmitter. If only one batch of neurotransmitter comes out, you get a small response, more neurotransmitter, more response. So here we are with our ligand-gated channels that I talked about earlier. The neurotransmitter is released from the presynaptic axon bulb and it hits this channel. Now this one could be a glutamate channel, right? An NMDA channel, could be a nicotinic acetylcholine channel. They're both sodium channels. So when they open, sodium comes in. And what happens here then is you get depolarization here because sodium's coming in. And that depolarization spreads along the membrane laterally. That's the passive spread of the depolarization. And the reason that's happening is you've got all these sodiums coming in. What are they going to do? They're going to find potassiums there. And charge repulsion, potassium moves away. And then that depolarization here, you can see -50, gets spread down the axon or the membrane. Now you can see, although it's spreading down, it's getting weaker as it goes, right? Here, the excitatory potential here, our EPSP, excitatory postsynaptic potential, is -50 millivolts, come down from what, -70, right? But it's decreasing as you get further and further away from that synapse because you've got that leaked potassium that's diminishing it. So EPSP, excitatory postsynaptic potential, is when you've got an axon releasing neurotransmitter. It's activating the ligand-gated channels, producing an excitatory potential here. Excitatory is always sodium. So you've got this excitatory potential that spreads from this area across the membrane, diminishing as it goes because of the leaked potassium. The opposite then is an excitatory or an inhibitory postsynaptic potential. So the other major type of synapse in your brain are the glutamate, or I'm sorry, the GABA synapses in the brain. They're going to release gamma aminobutyric acid, GABA, which activates the GABA receptor, which is a chloride channel. Chloride then comes in, and since you've got a negative charge coming in, it becomes more negative on the inside. So that's your hyperpolarization, or it's going to be inhibitory because it's going to inhibit the depolarization of the membrane. So let me just go over that again. So if this is an inhibitory synapse, typically you've got GABA being released, activating a GABA receptor. That allows chloride to come in, and then you've got hyperpolarization of the membrane. It goes from -70 to maybe -80, and it becomes a less excitable neuron. So this is our graded potential then. It's just, you know, the amount of depolarization depends on how much neurotransmitter is released. And it's one EPSP is really small. It's not going to excite the neuron that much. You need multiple of them to get together. So let's see that in the next slide here. So again, this is just cartoonish, but you've got a neuron here interacting with the cell body, the soma of a neuron, of another neuron, depolarizing to -40. So from -70 to -40. And that spreads across the membrane, diminishing as it goes because of that potassium leak. So, the way you get an action potential in this neuron is if that depolarization makes it all the way to the axon hillock. So if it makes it here, then an action potential starts and it goes all the way to the end of the axon, because of that the all or nothing principle of the action potential. Talk about that in a second. Okay, so this is your graded potential then. And typically a neuron's got thousands of axons interacting with it on its dendrites, on its cell body, everywhere. So what the cell body is doing then is taking all these inputs that are coming from other neurons and adding them together. Here's an example of, you've got a neuron here producing an EPSP and a neuron here producing an EPSP. They travel across the membrane again, but they add to each other, and you get enough depolarization here to initiate an action potential that will then go down the axon. Okay, so that's spatial summation. That's a summation of two different depolarizations. So again, your cell body is summing up all of its inputs, and it's got thousands of them. Some of them are excitatory, some of them are inhibitory, and it's taking the average basically of all that electronically. When that reaches the axon hillock, which is this area, it will produce an action potential. Now, if this neuron gets an awful lot of excitatory input, it's not going to get a bigger action potential. It's going to get more frequent action potentials. So the output here of this neuron is in terms of frequency of action potentials. The more stimulation it gets, the higher the frequency. More inhibition it gets, the lower the frequency. So that's more or less how neurons are working. Questions, comments? Okay. All right, so propagation of the action potential. How does the action potential go down the axon? So action potentials are all or nothing. Whenever when this part of the membrane gets stimulated, all the sodium channels in this part of the membrane open, and the action potential peaks there. So it's an all or nothing happen. It's as strong as it can be for any particular action potential. Okay, so you get depolarization here in this patch of membrane. Again, that depolarization is going to spread horizontally across the membrane, really in both directions, but if you're starting the axon hillock, it's only going in this direction. So that depolarization spreads across the inside of the membrane until it reaches the next node, let's say, the next part of the membrane here. And now the sodium channels here start to sense the depolarization that is occurring over here. And, oops, and they will open. And when these are opening behind it, these are repolarizing because of the potassium. And then when this depolarizes, it spreads across here and opens these. So it's just like dominoes falling down. Let me see if I can show you that animation, because I think it's somewhat interesting. Stop sharing. Share window screener tab. Share. Sharon's Karen, right? Okay. Oh, here's our action. Can you see this? This new share, people? Anyone?
Yes.
Yes.
Yep.
Okay, so here's your action potential. Sodium channels open. Come on. Sodium comes in, it peaks. Potassium channels open and it comes back down. So there's your action potential. Let's see it again just for the pure hell of it. Okay. So now here you can see that happening in each part of the membrane as the action potential passes down the membrane. So it's just like dominoes going down. This depolarizes, causes passive spread. This depolarizes, passive spread. This depolarizes, passive spread, and it goes down the axon that way. Okay, now where it gets interesting is when this is just a neuron with no myelination. So this is basically a slow neuron. What really speeds this up, and I'll go back over this more in a second. I just want to show this animation while we're here. What really speeds this up is myelination. So here between the different parts, we're going to call them nodes now. You've got all this insulation. What does that mean? That means here, what was decreasing this spread was our leak potassium, right? So it's not, if it's minus 80, or I'm sorry, plus 50 here, it's not going to be plus 50 here. It's decreasing as you get further and further away. With this myelination, you don't have any leak potassium anymore. So when you get depolarization here, it immediately goes all the way down the line to the other side of this myelination. Okay, this is called saltatory conduction. And again, that's just electrically, sodium comes in here, positive charges coming in, pushing these potassiums away, and then that depolarizes on the other side of the thing to the next node. Let's look at that in our PowerPoint now. Oops, that's not doing it. Okay, so here's myelinated neurons. So this myelination is put on by, in the brain, oligodendrites, in the body, Schwann cells. Basically, it's just a bit of their plasma membrane that goes out and wraps around the axon over and over and over again. So you've got many, many layers of phospholipid bilayer, and that provides insulation then. And so when you get depolarization here, that spreads all the way down here. So your action potential is, instead of going, you know, click, 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 click from one end all the way to the other, goes from here to here to here to here to here, and then it's gone. And that makes the action potential transmission a whole lot faster. In fact, you really can't live without it. You know, there are a lot of lysosomal storage diseases that end up causing demyelination, like Tay-Sachs disease and a lot of Krabbe type diseases and things like that. So this myelination is critical for life. Okay. That's about all I have to say about chapter 2. Let me stop my sharing. Any questions about that before I go on to chapter 3? I think I'm going pretty fast. I hope that's okay. I'm thinking I can push you guys harder than my undergrads. Are we doing okay? Give me a yes or a no. Everyone, give me raise your hand if you're good. Thumbs up. No. Okay, good. Everybody, most people are good. All right. In that case, I think you people think you're so smart. Let's move on and do more. How's that? So you should have said you didn't know, and then we'd go over it again. Sorry. I have to joke. It's what I don't get paid enough to not make jokes. I'm just kidding. All right. So chapter 3. Okay. So one thing. Let's see. Do I have any announcements? Oh no. Announcement 3. Oh, one announcement I wanted to make. I forgot at the beginning. I want to tell you there was a question on the quiz that was definitely wrong. It was like, if you put red blood cells in 35 milliosmolar solution, what's going to happen to them? And the answer was they're going to shrink. Now, the reason that was that way was it was a typo. It should have said 350 milliosmolar, and then it's going to shrink. And in 35, it's going to explode. So I gave everybody credit for that, just so that, you know, I'm sure everyone got it wrong because it wasn't possible to get right. So I gave everybody credit for that in the end. Okay, anybody have I see hands are still up. Do you guys have questions, or does it just not go down? Let me see what my chat says. Okay, my chat is telling me how dopey I was earlier on. Okay, we'll leave that open. Okay, so let's go on to chapter 3 then. And what I want to tell you right off the bat with chapter 3 is I really don't like the way this book organizes chapter 3. Chapter 3 is about cytoskeleton, and it starts out okay, here's what actin is, and now let's do skeletal muscle contraction. and then it goes back to polymerization of actin. So it's like, it's going from just saying this is what actin is and going to this really specific example of what it's used for. And then it goes back and does all this general stuff about actin and how it's polymerized and all this stuff. And I don't like that order. It feels kind of out of place. So I've reordered it. So we're going to start out talking about actin microfilaments, how they're polymerized, how they work, how they're used in cell crawling, and then we'll go into skeletal muscle contraction, cardiac contraction, and then we'll do smooth muscle contraction. And that's all microfilaments there. It's all actin. And then the next section is intermediary filaments, which is kind of short. There's not a lot to say about them. And then the next segment is microtubules. So, and we'll talk about movement for all of this cytoskeleton stuff. Okay, so let us move on. Oops. Gonna hide my presenter view. Okay, cytoskeleton. What is the purpose of the cytoskeleton? Number one is going to determine the shape of the cell. Here's 4 examples of different cells. They all have extremely different shapes based on their function, their purpose. And the reason why these are different shapes is because they're cytoskeleton. Keep in mind that the plasma membrane is an extremely unfavorable energetic thing, okay? The plasma membrane would not be this shape if there wasn't cytoskeleton holding it together. It would collapse into vesicles, right? An extended phospholipid bilayer is not energetically favorable. Without that cytoskeleton holding together, it would collapse into vesicles. So what's holding the shape of the cell then is the cytoskeleton. Another thing that cytoskeleton does is, when I was taking biology way back in the early 80s, frankly, we were kind of led to believe that a cell was a bag of water with stuff sloshing around in it, That's definitely not true. That would be an incredibly inefficient way to run a cell. That's no way to run a cell. So anyway, a lot of part of what cytoskeleton is doing too is kind of compartmentalizing things. So it'll hold enzymes all in one place so that, substrates will be near the enzymes and things can be really efficient. It's also going to be holding and moving organelles around and stuff like that. Protein RNAs can be compartmentalized. I think my favorite example of RNA being compartmentalized is early embryo development, especially in the xenopus. It's like there's an egg cell, and the egg, you would think it's one cell, it must be uniform, but it's not. The bottom of the egg cell has got all these messenger RNAs that aren't in the top. And once the cell starts to divide, that determines endoplasm versus ectoplasm. So, even messenger RNAs can be attached to the cytoskeleton, so they're not floating around in the cytoskeleton, in the cytosol. Again, remember, nothing just happens accidentally in a cell. Everything is controlled. The cells where it happened accidentally are 4 billion years old and they're dead already, right? Because all these things had to get more and more efficient as the competition for survival went on at the cellular level. Okay. That's one thing it does. It holds the internal environment together, holding things where they need to be. Also, it's going to provide for movement. Anything moving around in the cell is going to be interacting with the cytoskeleton. When the cell itself moves, whether it's a cell shrinking for contraction or crawling across the surface, that's all cytoskeleton. So cytoskeleton is really important for movement. It's kind of like your own skeleton, right? If you didn't have a skeleton, you couldn't have movement. You'd sit there and quiver. Wouldn't that be uncomfortable? What else? Oh, really important during mitosis, especially microtubules, right? The microtubules from the interphase cell are going to depolymerize and then repolymerize in the form of the mitotic spindle. And that's key for separating sister chromatids and making 2 cells from one. And then also for the division of the cell after mitosis, which is now going to be microfilaments and myosin. So the microtubules during anaphase of mitosis will stretch the cell out, and then a belt of actin forms in the middle of it and crunches down until you have 2 cells. So anything involving movement is cytoskeleton, and that's what we're going to talk about. Okay, so cytoskeleton is made up of 3 different elements. We've got microtubules, which are the biggest of them and the stiffest, really. They don't bend as easily as the other ones. Then you've got microfilaments, which are actin. These are made of tubulin. These are made of actin. And these are kind of like more rope-like, you know, they're kind of like a rope-like structure. And then you've got your intermediary filaments, which were somewhere in the middle. And again, we're not going to talk a lot about these. There's not a whole hell of a lot to say about them, except they're structural. These are both involved in movement, so we end up talking about them a lot more. These are directional. You've got 2 different ends to this molecule, 2 different ends to this molecule. These are not directional. So these are just going to be structure. Okay. Actin. So we're going to talk about actin first. So we've got a lot of different actins. So we've got muscle actins that are going to be used for contraction. You've got different isoforms for skeletal, cardiac, and smooth muscle, vascular, or whatever. And then you've got more non-muscle actins, and these are in muscle cells as well. They're just the only thing in non-muscle cells. So you've got beta and gamma actins in non-muscle cells, gamma enteric in smooth muscle of the viscera. So that's pretty specific. But so anyway, your alpha actins are in for contraction, and your beta and gamma actins are more structural in all cells. So, you know, whenever you're doing an experiment where you want to measure whether a specific protein is increasing or decreasing under certain conditions, you always have a standard there. Oftentimes it'll be actin, because all cells will have that actin at the same concentration, holding the plasma membrane up or whatever. So that's what these non-muscle actins are doing. They're more in the cortex of the cell, holding the plasma membrane in place. All right, structure of actin. So actin are small globular proteins, and it's got a binding site in the center for ATP or ADP. It will hydrolyze that ATP into ADP once it polymerizes. So the ATP is important for the polymerization of the actin into filaments. So this we call G-actin because it's globular. This we call F-actin because it's filamentous. But anyway, this actin monomer will bind ATP and then polymerize. And after it polymerizes, it'll hydrolyze that to ADP. And then this structure can fall apart if it's not stabilized somehow. So we'll talk about proteins that stabilize this. But the bottom line is that, you know, you need ATP to polymerize this. Once it's polymerized, it will be more or less stable. Now, it doesn't look like it here. It's just globular circles, but there's a difference between one end and the other end of a microtube of a microfilament. One end will be called the plus end or the barbed end, and the other end is called the minus end or the pointy end. Now, it doesn't have anything to do with plus and minus, doesn't have anything with charges. It's just like this end versus that end. And the reason they call them that is if you took a myosin head, let's say this is myosin. Watch me draw. Okay. You took a myosin like this and added it to the actin, it would bind to the actin like this, and the tails would head out this way. So this ends up being the plus end, and then this ends up being the minus end. So this is like the barbed end because this part of the myosin is sticking out. And you can read about that in your book. They talk about how you take a certain myosin, you cut it with papain, and it cuts it off here, so the long tail is gone, but it still has a short tail. And when you add it to the microfilaments, you can distinguish plus from minus end. So that directionality is really important because when we start talking about movement, we're going to talk about myosin walking on that in one specific direction, not the other. So the directionality is really important. Okay, how does it polymerize? This is in vitro. You can learn something about this, about polymerization in vitro. Basically, you put a whole bunch of actin together along with ATP and magnesium. Magnesium is always involved with ATP. It helps to neutralize some of the negative charges on all those phosphates. Helps to coordinate that ATP molecule in the enzyme that it's in. I always see magnesium with ATP. So anyway, you get magnesium and ATP together with a whole bunch of monomers. It's going to take a little bit to get the reaction started. They need to nucleate. Once they nucleate, boom, they can polymerize really quickly. It's kind of like when you take distilled water out of the freezer and it's still liquid, even though it's like minus 20 degrees. You tap it, it nucleates one little ice crystal and the whole thing freezes. It's just like that. Once you get nucleation, it polymerizes really quickly. And then once it gets to a certain length, you find out that one end, the plus end, preferentially adds monomers, and the other end preferentially subtracts monomers. And that's because the plus end will add at a lower concentration than the minus end. So you get to a point where the concentration of free actin ATP drops, and it's still high enough to add on the plus end, but it's not high enough to keep adding on the minus end, so this decreases. So this is treadmilling, okay? And we'll see this in a cell when it starts to walk too, the cell crawling. You're adding at one end and subtracting at the other so that it's kind of like changing, but it's not changing the length. That's treadmilling. Okay, that's in vitro. In vivo, this is all going to be controlled by specific proteins. Now I want to change my share here, and with any luck, I can do this without the world collapsing on me, as it tends to want to. Okay, so here's the notes for lecture 3. So these are the proteins that are involved. Can everybody see this, my notes?
Yes.
Yep.
Yep.
Okay, good. So these are the proteins that are going to be involved in polymerizing actin when it starts to change. One reason it might change would be cell crawling. We'll see that in the next class, how you get a lot of polymerization, depolymerization of actin during that process. And I'll show you, I'm going to show you some really cool animations next of showing how this actually works. But anyway, the things that you need to know about this are, number one, CAPZ. So CAPZ is a protein that's going to bind to the plus end and stop it from depolymerizing. So again, if you, in vitro, if you stop adding ATP, it's all going to fall apart because it's going to hydrolyze that ATP and become unstable. Same thing's going to happen in vivo, except instead of them falling apart, that plus end is going to be capped, so it's not going to depolarize. And that CAPZ can also bind to other proteins. So now you've got your microtubule attached to another structure. Thymosin beta 4. So interesting thing your book said, which I didn't know, which is kind of interesting. There's a lot of free actin in cells. There's a lot of free actin in cells, probably because they need to change their microfilaments quite often. So the concentration of actin in the cell is high enough that it should spontaneously start to polymerize. And that would just be a mess if you know, microfilaments are just polymerizing randomly all over the cell. So thymosin beta 4 there is to sequester these monomers, so you're not getting that. That's not happening, and it's just going to hold them there until the cell's ready to change. Profilin then is going to promote the exchange of ATP for ADP in the actin monomers and transfer them from thymosin beta 4 to the open plus end. So when it is time to polymerize and get longer, profilin is going to grab the actin from thymosin beta 4, exchange the ADP for ATP, and then bring it to the plus end. Once it gets to the plus end, Formin is the enzyme that's going to grab that actin from profilin and put it on the plus end. And it's really cool. It almost it looks cartoonish the way it actually works. It's friggin' hilarious. The Formin's got two arms and it's grabbing actin and putting it down, putting it down, putting it down. You'll see in the next in the animation. It's really cool. Oh, also a little note here. Formin is given a different role here in your text on page 78. Ignore what they say about it. I think they're wrong. And when you see the animation, you'll see it makes a lot more sense the way I'm telling it here. Formin's basically sitting there on the plus end, grabbing actin monomers from profilin and adding them to polymerize. Another part that's important here is when you're polymerizing at one end, you're typically depolymerizing at the other end. That's where you're going to get your monomers from that can be pushed forward for the polymerization. Especially important in amoeboid movement, where you've got the gel to sol phase transition. We'll talk about that in the next class. So ADF cofilin is binding to the minus end and depolymerizing from the minus end. And then gelsolin is going to make that faster because gelsolin cuts in the middle of the strand, and that produces a whole lot more minus ends, and then ADF cofilin can depolymerize faster. So this is our basic process here. You've got CAP-Z binding the plus end. When CAP-Z comes off, then Formin can bind, and profilin brings monomers over to Formin, and it polymerizes. And then at the other end of the microtubule, you've got ADF and cofilin and gelsolin depolymerizing. Now, another thing that's going to happen, especially in the lamellipodium of a crawling cell, is you're going to have branching going on. And I've got a cool animation for this too, so I'll tell you about this, then we'll look at these animations. So branching is based on three different proteins here. CDC42 is a small monomeric G protein. So if you've had cell biology before, you've probably heard of the protein RAS. RAS is an important signal transduction G protein that's activated by tyrosine kinases. CDC42 is part of the Rho G protein. So RAC, Rho, CDC42. I'll talk about these three again in the next class. But anyway, the G protein gets activated by a tyrosine kinase. That G protein then activates WASP, which is going to grab ARP23 and stick it on the side of a microfilament, and that becomes a nucleation point for a new branch to go off the microfilament. So the previous mechanism we talked about is all just making straight chains of actin. Here, this WASP grabs a new nucleation site, sticks it on the side of actin, and then you get a branch coming off that actin. So let's watch these excellent videos. I think you've probably learned by now that I absolutely love videos. I think it's just so much easier to understand things when you can see them this way. Bum, 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 bum. I'm getting pretty good at this Microsoft Teams stuff, if I have to say so myself. Oops, that's my cell phone. Ignore it. That was not what I wanted. Window. Where is it? Where is it? Where is it? Chrome tab. Nope. Window. What the heck? What the heck? All right, let's do this. Oh, sorry, I was just checking my email there. I'm just kidding. Where are we? All right, let's stop sharing. Maybe that will help. Share window. There we are. Okay. Okay, so this is Formin-mediated actin nucleation and filament assembly. I'll let you watch it, and then I'll go through it again and explain it in case it didn't make sense to you. I'll try and shut up. Okay, so I'm sorry, I have to say something. We're activating CDC42 here, and here comes Formin. All right, let's go through this. Let me let me talk because it's what I do for a living. Let me talk about this as it's going through. So what's going on here? This is activation of the process. So a GEF is going to be a guanine nucleotide exchange factor. Here's your Rho GTPase, which is CDC42. Okay, and the GEF is going to activate the Rho. Okay, once that becomes activated, it's going to cause Formin to come over and start a new chain. So Formin now is binding to that CDC42, I believe, there. And now it's going to start bringing actin monomers in. It's going to bring them in and nucleate a new fiber here. And then start adding monomers. Now it's going slow here because we don't have profilin involved. Now here it's a lot faster because you've got profilin. And if you notice, when it slows down, it'll be easier to see. Profilin is binding to Formin and bringing it in. So that interaction of profilin and Formin helps to bring the actin monomers in faster, and then the polymerization can happen faster. Okay, so that's your formation of just a straight, just a single chain of F-actin or a microfilament. All right, so let's see how branching works now. Okay, I have to change my share. Oops, not that one. Stop sharing. Change my share. Come on. Okay. Okay, sure. All right. So here's your branching now. So ARP2 and ARP3 look a lot like actin, and that's what's going to allow them to nucleate a new chain off the side. So here's WASP. You can tell by the name it's involved in a genetic disorder. So it normally auto-inhibits. This is classic biology. Along comes CDC42, takes up that site and releases the other site. Now this site, the other domain. Okay, so this is showing when it's not activated, it doesn't interact with 2.3. When CDC42 comes along now, boom, exposing that domain, which then binds to ARP23. Okay, now we're ready to branch. Okay, so that's going to branch off now to give you branched microfilaments. And that's it. Okay. Okay, that's about all I want to talk about today. Any questions or comments about that? Are we all good? Can we see your faces so we feel like a class here? Here we are. Hannah's got a bad camera, so we're going to let her go. All right, guys, that's about all we have for today. Let's get together and do this again on Wednesday. How's that sound? Yay, Wednesday. All right. See you later, guys.
I have a question on,
Okay.
I guess about the exam. Is the exam going to cover chapter 2 and 3 now, or is it still chapter 2?
I think 2 and 3. We'll
Okay.
see how far we get through 3. We'll probably finish 3 by the end of next week, and then we'll have the exam on both. I'll
Sounds
change
good.
the syllabus to reflect that, okay?
Thank you.
All right. See you later, guys.